Right, yes, so how many cat owners do we have in the room? Oh, it's so nice that they let you out. Okay. So those cat owners in the room, your cat probably has had or probably will have at some point in its life a feline coronavirus. Feline coronavirus 1 and 2 are very common all around the world, but typically if your cat gets it, you will never know because it will not show any clinical signs whatsoever. Sometimes there'll be very mild diarrhea, but you're probably still not going to notice it. So if clinical signs are present, very mild, but it is very efficient at transmitting via the fecal-oral route, so it's a very successful virus, it's everywhere. But it's not something you should ignore. So in a subset of cases, this virus mutates once it's already in the host and becomes a more pathogenic version of the virus. And this causes a range of clinical signs that are kind of summarized in the figure there, but we broadly cluster those into 3 different kind of groups. So we have a dry version of this, where basically lesions form in different organs around the cat and cause all kinds of weird and wonderful, or not wonderful, symptoms in the cat. We have the neurological form, and you see things like seizures and ataxia, and general typical neurological signs of something going on in the brain. And then we have the wet FIP, which is the most recognizable one. They kind of look like they're pregnant, they swell up a lot, and this is the version that kills the cat the fastest, because unfortunately all of these are fatal without treatment. And the treatment is with antivirals, very similar to how we treat SARS-CoV-2. These are very expensive, and not available for use in cats in most countries. There's only a handful of countries where you can use these in cats. Luckily, the silver lining to this is that once the virus has turned into this horrible form, it's not very good at transmitting. So it's switched its tropism from the enteric tract to macrophages, and it's no longer shed as efficiently in the feces. So it's really only a problem for the cat that has it. That being said, what is happening in Cyprus? You may have seen some of these headlines last summer of a large outbreak of this more pathogenic form, which we call FIP, which has been killing a lot of cats in Cyprus. This doesn't really make sense with what we know about this virus. Now, when we have a look at actual numbers out of Cyprus, this doesn't look that staggering. I don't know if you can actually read on the screens there, the scale on the plot there. It maxes out at about 37 cases in a month, which is not that staggering. And you can see that we had this one big wave, and then it's kind of calmed down a little bit, and then we see the start of a second wave towards the end of the plot. Again, this doesn't seem very staggering, but something we need to remember about Cyprus is that most of the cats in Cyprus are strays. So no one is taking most of these cats to the vets, they're not being tested. So to see what the real impact of this is, we need to look a bit closer at the stray populations. So most of the cats in Cyprus live in this kind of setting. They call them shelters, but they're not kind of shelters in the way that in this country we'd refer to shelters. They're not necessarily looking to rehome these cats. They're looking to feed them and look after them and give them the best life that they can. It's very rare that they're sort of efficiently rehoming these cats. They tend to just spend their lives there. When we talk to these shelters about what's been happening over the last year, they report losses of between 30 and 40% of their cats with FIP symptoms. And given that Cyprus, well, the last estimate before all this was that Cyprus had about 1.5 million cats, that I'll let you do the depressing maths on that yourself. So in the past, I've designed these tiled amplicon sequencing schemes for a few different viruses. You may be familiar with this as one of the methods that was used to sequence SARS-CoV-2. And the general idea, very briefly, is you're designing amplicons that when put together later will cover the entire genome. And you do this in 2 separate reactions. So the whole genome gets amplified in 2 reactions, put it back together, sequence it. So I have experience in designing these. So last summer, I was asked to design one of these schemes. And I said, sure, that shouldn't take long. So we managed to get this scheme working on Friday. So it did not go very smoothly. This is not because I suck. I promise it's not. The method to design these schemes relies on access to high-quality genome assemblies for the virus you want to sequence, and they should be as closely related as possible to the outbreak strain. We do not have any of those. There are a few genomes available for feline coronavirus in general, 85 at the time that we were trying to do this. These are very diverse, they're from all over the world, they're different serotypes, they're from different time points, they're very, very diverse. Most of them are not feline coronavirus 2. The one thing we know about this particular virus is that it is serotype 2 when I started this project. So it'd be helpful if there were more feline coronavirus 2s in the database. They're not annotated by disease status. Now, as I say, the mutation within the cat that causes FIP, it's a de novo mutation, so it's not the same in every cat. So if you're trying to do a multi-sequence alignment and you don't know the disease status, you can have all kinds of weird and wonderful deletions going on that are going to mess with your multi-sequence alignment. So it'd be really helpful if they were annotated by disease status. So as you can imagine, this did not go well. It is going well now. But it wasn't when I did up these slides. So most of these slides aren't going to be about the scheme itself, but about what we learned along the way. So several regions did amplify quite well. And we also designed an additional amplicon that would span pretty much the entire spike gene, because that is where most of these mutations that cause FIP tend to occur. So we sequenced with the Q20 kits, so the previous generation of kits, on our GridION, and did super accurate base calling. So having a look here at what came out of this, so these are some of the regions that amplified really well. If we have a look on the left-hand side here, we can see that the samples with the green arrow that are highlighted in yellow, these are our Cyprus samples. And interestingly, they're in feline coronavirus 1. So the one thing I knew about this virus was not true. It was not a feline coronavirus 2 at all. So that clusters nicely with feline coronavirus 1. The other end of the genome similarly clusters with feline coronavirus 1. But then we look at the spike gene, and it's not with feline coronavirus 1 or with feline coronavirus 2. It is sitting with the canine coronavirus 2s. So that was a bit of a surprise, and explained why none of the primers in that region worked when we were trying to do the scheme. So this is not too unusual. This group of coronaviruses does this stuff a lot. They shuffle around the spike, they swap spikes a lot. The existing strains and serotypes that we have came about through various spike recombinations in the virus's evolution. And they also do other things, so we can see that canine coronavirus 2 there, over time evolved into a porcine virus that infects the enteric tract. And a small deletion, well, quite a large deletion actually, in the spike gene of that virus created a respiratory virus in pigs. So the spike gene is quite important for things like tropism. It changes things about this virus. So having spent some time very carefully piecing together a complete virus to have a look at the whole genome, we can see that the recombination region is very tight around spike. So whereas feline coronavirus 2, which was similarly formed with feline coronavirus 1 recombining with canine coronavirus 2, that had a much broader recombination, so much more of the genome recombined in that case. In this case, we're very tight around the spike gene. And we can also see that the majority of our samples had a deletion, which is almost identical to that pig deletion that changed the tropism in pigs. So that was quite interesting as well. So why does that matter? I've just said this has already happened before. Feline coronavirus 1 has recombined with canine coronavirus 2. We got a new feline coronavirus. It's, why should that change anything? So the closest relative to the spike, and it is quite close, is a pantropic canine coronavirus. So similarly to feline coronaviruses, canine coronaviruses typically we don't actually need to worry about. However, this one is a nasty one. This one kills dogs. So this one, again, 100% mortality unless you've got treatment. And what we think is happening here is that the spike is allowing the feline coronavirus to have much broader tropism. So it's not just in the macrophages or just in the enteric tract. It has a broader tropism like this canine coronavirus. And we do see that this is some staining of the colon of a cat with the new strain of feline coronavirus that we're calling FCoV-23, compared to an FIP case where the virus is just in the macrophages and not in the enteric tract. So unfortunately, we're also seeing this in the feces, that it is actually shedding in the feces. So do we need to worry? Yeah, unfortunately it is in other countries. It has been confirmed in Belgium, and there are some clusters of FIP showing up in other countries as well, although we haven't tested them yet to see if it's caused by this. Unfortunately, Cyprus exports a lot of cats, primarily to the UK and Germany through what we call freedom flights, or rather what they call freedom flights, for people wanting to rehome cats from Cyprus into their country. So there's a lot of movement of cats going around. We do not know if this will spread as well in other countries, though. Cyprus has got a very high density of cats, and the animals in Cyprus have probably got exposure to the parental strains, so that might actually be slowing it down in Cyprus. So we don't know how this is going to behave in other countries. We don't know what else carries it in terms of other species or fomites, and we don't know if there's a broadening of transmission routes. So there's a lot to find out, and we're working on it. So just to summarize why nanopore sequencing was so useful in this, the long amplicon of spike was an immediate answer to what was going on. We could look at the structure of the protein from that, we could see the recombination breakpoints from that, and we can use that to reliably diagnose the feline coronavirus. There was a lot of trial and error in creating the scheme that does now work, and having the in-house sequencing for that was really useful. And once this scheme is finalized, of course, we'll be able to send this out to be used wherever there is a nanopore sequencer, which, as we know, is pretty much everywhere these days. So yeah, to summarize, big nasty virus. Hopefully, it will not be as successful in other countries as it has been in Cyprus. So I'll just thank everyone who's involved in this project. There's a lot of people. There's actually even more people than are on here that have sort of joined in the team after this to look at things like immunology and vaccines. And also my cats on the left and my supervisor Christine's cats on the right. So also thank you for listening.